So I figured we could spend a few minutes sort of deconstructing the history of Mendelian genetics. Briefly, Mendel is this monk in what is now the Czech Republic who notices that there is these pea plants that he likes to grow in the garden that present with two types of seeds of peas. Some are green, some produce yellow peas, right? And like many other scientists, amateur scientists at the time, he's interested in how traits are inherited or passed on by organisms, both animals and plants. And he's very aware of the prevailing theory of the time, which is the theory of blending inheritance. But he keeps getting in his garden these plants that produce either green or yellow peas, nothing in between. So that was an observation that caught his eye, because according to the blending theory of inheritance, after a little while, all the plants should produce this greenish yellow or yellowish green, because traits blended generation after generation. But no matter what, Mendel keeps observing this dimorphic trait, right? So some plants produce green peas, some produce yellow peas. Now, this idea that he was the first observing this is something that may come across from books and sources, that he came up with a particulate theory of inheritance because he started seeing these things. People had seen this before, obviously. It's just that he put numbers to it and decided to investigate it in further detail. When thinking about why it might be that these traits weren't blending in progeny, there were a number of possibilities. Maybe the green pea plants did not fertilize with yellow pea producing plants and vice versa. So the reason why there were these two subpopulations in his garden is because they were not mating with one another. So that's a possibility. Another possibility was that pea color itself is not inherited, right? So it's something that is produced generation after generation from environmental inputs, like the amount of sunlight that a plant receives, the amount of water, the quality of the soil, right? Some things that we may not have so much control over. So Mendel used a concept that he didn't invent. That's another misconception that may get a little confusing in textbooks and whatnot. He uses selective breeding, but he didn't invent that. Selective breeding had been invented by humanity from agriculture and breeders thousands of years before, right? Essentially what he says is, well, if I can control which plants made with which other plants, then I can start figuring out whether this dimorphic character of pea color may have something to do with the fact that green pea producing plants don't fertilize with yellow pea producing plants, and vice versa, and so forth. So obviously the challenge with freely fertilizing plants, like when you go in the wild or you have a garden and plants are mating with each other by themselves, is that you can't control who's mating with who. But he had developed this ability of being able to control which plants a given plant were mating with by using a brush and taking the pollen grains from one plant and carefully introducing them into the ovaries of a flower in a different plant. So that's how he could control matings, and he could carry out this, what we would call nowadays controlled genetic crosses, right? So he does that, and the first thing that he decides to do is to make sure that he would have a line of plants that produces green peas all the time and a line of plants that would produce yellow peas all the time. So he could start with pure plants that would producing nothing but one type of pea color, right? And he does that through self-fertilization of plants. He keeps self-fertilizing these plants to themselves until they will produce nothing but progeny with green peas or yellow peas. And he calls them pure breeding lines. Okay? Then, now knowing that these plants cannot pass on to their progeny anything but the green pea trait or the yellow pea trait, trait respectively, he decides to cross them to one another. And he carries out one of these crosses between pure breeding green pea plants and pure breeding yellow pea plants. And he calls the progeny hybrids, right? Because he knows that these progeny is coming from two different pure breeding lines. And the first thing he notices is that there are no plants producing green peas. All the plants produce yellow peas. So what happened to the green peas? Right? There were a number of possibilities. Now, I used to ask a question in this class previous semesters. I would ask the question, and everybody would tell me, oh, because yellow is dominant to green that is recessive. I know you guys know that. But at the time, I'm sure Mendel may have thought of other possibilities. Maybe yellow was, quote unquote, destroying the green, right? Or maybe green was hiding. Or maybe green was never being passed on if a green pea pure breeding line was crossed to a yellow pea producing pure breeding line, right? So there were a number of possibilities. So how could he find out what was going on with these hybrids and what was going on with the green trade, with the green peas trade? So to find that out, he decides to now self-fertilize these plants that had been produced by crossing the original pure breeding lines. Excuse me. And by the way, let's throw in some nomenclature. So this original generation with the pure breeding producing lines was called the P generation for parental, or F0. And then their progeny was called the F1 generation. So he decides to self-fertilize F1 plants to see what's going on with them, to see what's going on inside of them, if you will. And what he notices is that every time he does that, he obtains progeny with a very distinct feature. The progeny would have mostly yellow pea producing plants, but some green pea producing plants. And when he counts, he notices this interesting ratio of 3:1. 3 plants producing yellow peas for every 1 plant producing green peas. It didn't happen with one plant from the F1, it happened with all of them. So whenever you see something like this, as a scientist, we would these days, and he did back then, ask yourself, is this some sort of fluke? Or is there something super special about pea color as a character, but you wouldn't see it with other physical characteristics of these plants? So the next thing he does is he tries the same approach of generating pure breeding plants and then seeing what happens when he creates these hybrids with 6 other characters. So we already spoke about the pea color, and then he tries 6 more for a total of 7 different characters. And all 7 of them show the same behavior. What I mean by that is there was a character that is dimorphic, so it comes in one form or another. So there were the character pea shape came in wrinkled and smooth or round. The character flower color came in purple or white. The pod shape was constricted or full, meaning a smooth surface or a rough one. The pod color itself came in green or yellow, and so forth. So these 7 different characteristics, all of which were dimorphic, came in 2 different shapes. And when he created pure breeding plants for either of the morphs in each character, when he crossed those, he would get an F1 hybrid generation that was 100% one of those traits. So 100% yellow peas, or 100% purple flowers, or 100% tall plants, and so forth. And then when he would self-fertilize the F1 plants from each of those, for each of those experiments, he would get always this very unique 3-to-1 distribution of traits, with one of them showing up 3 times as many as the other. So far, so good? Okay. Normally, when you start learning genetics, even in college courses, sometimes what happens is we go forward with what we already know about laws of inheritance, and then we start explaining some of these observations based on that basic knowledge. Here we're doing the reverse. We're kind of walking the path that Mendel walked and trying to figure out what was happening the way he did, and based on observations, and then from observations going through hypotheses and finally a model. So from these observations alone, he's already coming up with some like general conclusions that didn't apply to p-color only. They apply to all 7 characters that he was working with. First, he saw that some of the traits that sort of disappeared in the hybrid generation phenotypically, that they would not manifest physically to his eyes, were not destroyed or missing. They were just hiding of sorts, right? So he started naming some of those traits that he could see in the F1 hybrid generation as dominant traits, and the other ones, the ones that were in hiding, recessive. They were like in the background, right? And he knows they're hiding in the background because when he self-fertilizes those F1 individuals for whichever of these 7 characters, these recessive traits reappear in some, about 25% of the progeny. Another thing he introduces that we use to this day is a little bit of nomenclature. So it is tempting to say, all right, I'm going to name the green piece G and the yellow piece Y, but he introduces this nomenclature by which the recessive characters and the dominant recessive traits, and dominant traits would be named with the same letter, but the dominant trait would be a capital letter and the recessive trait would be a lowercase. And that is very convenient because now we know based on one letter what character we're talking about. So in the case of p-color, he wouldn't refer to this green P producing plant as G and the yellow P producing plants as Y. Instead, he would refer to the green. Oh, there you go, I had it right. Yeah, so the green, the recessive with a lowercase y, and yellow, the dominant, with a capital Y, okay? And then another thing that often, based on what we know today, we rush a little bit to think is that Mendel already spoke of heterozygote and homozygote individuals. Not yet. He didn't have this nomenclature at hand yet. So at the time, he was still referring to some of these things as pure or hybrids, right? To indicate that a line had the ability to pass only one type of trait or the ability to pass on more than one. Any questions so far? All right, here's where it starts getting a little more interesting. So, just like he did with the F1, to try and find out what each of these F1 plants had inside, he self-fertilized them. He did the same thing with the F2 plants, those plants that were produced by self-fertilization of the F1 and that were always showing up in this 3-to-1 phenotypic ratio of 3 dominant traits for every recessive one, right? So he self-fertilizes the F2 plants, and he notices that the plants with the recessive trait, again, we go back to the example of p-color, the plants with the green piece, the recessive trait, always produce 100% of plants with green peas. Right? So they had become pure again. Those green pea producing plants in the F2 were pure breeding. With the yellow pea producing plants, the story was a little more complicated. It turns out that 2 out of 3 of those yellow pea producing plants were hybrids. How did he know? Because when he self-fertilized those F2 yellow pea producing plants, some green pea producing plants would show up in the F3 progeny. So that meant that these yellow pea producing F2 plants had some recessive green in them. They were just hiding it. And then for every 3 yellow pea producing plants, there was 1 that was pure breeding again. So he sees these with the pea color, he sees these with flower color, he sees these with pea shade, with pod shade, with plant hive, and so forth. with each of the 7 characters that he's observing. He notices that in the F2, phenotypically, so from a physical characteristics point of view, there is 1 recessive trait for every 3 dominant ones. But then, in terms of their constitution, that's kind of the word he used, the distribution was 1 pure breeding recessive for every 2 hybrids for every 1 pure breeding dominant. And that's where all lights must have gone off in his head, because he recognizes this 1 to 2 to 1 distribution as something that he has seen before, and he had seen in the field of mathematics. Okay? And it goes something like this. He knew that the 1 to 2 to 1 is something that you can easily obtain from what is called the binomial expansion. And we'll walk through that. I don't know how long it's been since you guys saw this in any math class, but we'll walk through it together. So the binomial expansion is a concept in mathematics that applies to trying to predict what the outcome of certain events may be. So imagine that we have an event with 2 possible outcomes, and we assign to those outcomes probabilities P and Q. We'll use a coin toss as an example. When we toss a coin, the probability of getting heads, or H, is 50%, 0.5. The probability of getting tails is the other 50%. So that's one event, a coin toss with 2 possible events with 50% probability each. Okay? So we know that, he knew that, so far so good. Where mathematicians come in is when people started to try and predict what the probability of combined outcomes might be. So let's say that now you toss the same coin twice or 2 coins simultaneously, it doesn't matter, 2 independent coin tosses, and you try and predict the probability that you get either 2 heads, 2 tails, or a combination of 1 head, 1 tail, no matter which one's first. Okay? So to figure that out, that's when mathematicians come up with the binomial expansion. And the binomial expansion is basically the product of P plus Q times P plus Q. And when you distribute that using FOIL, you get PP plus 2PQ plus QQ. If we apply this binomial expansion to the example of a coin toss, the idea here is that the probability of 2 heads and tails, or 2 tails, you can calculate by doing 0.5 plus 0.5 times itself, and then you get this distribution of 0.25, 0.5, and 0.25 for getting heads twice, getting tails twice, let me do that with color coding, or getting 1 of each. Okay? And it's all in the math, all mathematical. And that is what Mendel recognizes immediately. That 1 to 2 to 1 that he knew from the binomial expansion is something that he recognizes in his F2 progeny that had 1 pure breeding recessive for every 2 hybrids for every 1 pure breeding dominant. So that's where, in his mind, the thought must have come that maybe the F1 is producing an F2 that is following that 1 to 2 to 1 distribution, because what the F1 is doing is like tossing a coin twice, right? And that the F2 is nothing but the combination of the outcomes of those 2 coin tosses. Now notice here that this concept requires something key, and that is the fact that progeny of any mating event would be the combination of 2 things. That's a big intellectual leap, right? Did Mendel know if there was combination of 2 things in plant fertilization? What do you think? Did Mendel know what 2 things might be combining during plant fertilization? I see some heads nodding no, but some nodding yes. What do you think he knew about plant fertilization, that there were two things combining?
It's from like genetic inheritance, because before Mendel there was a protocol. It's a mix of both parents.
Uh-huh.
And he believed that.
So the parents, but plants can mate to themselves. So, but what are they producing that needs to combine? We mentioned it a few minutes ago. Exact, the pollen and the ovules or the egg cells in the ovary of the flower. That's right. And in animals, it would be sperm cells and egg cells for animals with sexual reproduction. Right? Exactly. And Mendel did know that. So much so that he could control those crosses because he knew that he would brush off some pollen grains from one plant and introduce them into the ovary of another or into the ovary of the same plant for self-fertilization. So that's how much he knew that the next generation came from the combination of two things that the parental plant was producing. Does that make sense? So it wasn't a huge leap. We tend to think that, oh my God, this guy, where did he get the idea from? Well, it wasn't a huge intellectual leap to think the parental plant is tossing a coin twice, and then those coin tosses combine in progeny. He knew about that combination because he had to actually carry out that combination to do any of his experiments. He knew that he had to combine pollen grains with ovules to get the next generation. So now the question, in a way, became, could the formation of those gametes, of those pollen grains and ovules, be like a coin toss? So he starts playing around with that idea that the F1 plant is a hybrid that carries a coin of sorts, right, with two faces. One is the ability to produce yellow peas. The other one is the ability to produce green peas, right? He knows that those two traits exist in the hybrid plant because he can see both of those traits in the progeny. So he knows that one of them is hiding, but they're both present there. And he starts playing around with this idea that maybe they're in that parental plant not in equal quantities necessarily, but with equal probability to be present in the gametes produced by those plants. In other words, that F1 parental plant had the lowercase y, the green pea trait, and the uppercase y, the yellow pea trait. And that when these plants were producing gametes and ovules, plants produce both, they're hermaphrodites, every time that they produce a pollen grain or an ovule, they would be like tossing a coin, and each of those pollen grains or ovules would get one of these two faces of the coin. They would get either the capital Y or the recessive Y, the lowercase y. And combining the pollen grain and the ovule was, in a way, like combining the outcomes of two coin tosses. And that's how he could obtain one pure breeding recessive, meaning nothing but lowercase y, for every one pure breeding dominant, nothing but capital Y's, for every two hybrids. Did that make sense? This is one of these cases in which an observation leads to a hypothesis based on some prior knowledge, same way that science works today. So we make observations, and using some prior knowledge, we try and make or formulate a hypothesis might explain those observations. And now we need to, quote unquote, test those observations. So he formulates the hypothesis based on those F1 self-fertilizations. That's the inspiration for his hypothesis. But now he can contrast, as we would say in technical terminology, his hypothesis with separate observations. And in this case, some of those separate observations came from his own previous experiments. Namely, could this model explain what he saw in the F0 crosses, in the F2 crosses, and so forth, right? So let's take a look. When it came to the crosses with pure breeders, whenever he was crossing pure breeders to themselves, it's not like the coin toss model goes out the window. According to his model, these plants still had a proverbial coin in them. It's just that it was one of these loaded coins with two faces being the same, right? So the pure breeding plants had a coin with two heads, and the pure breeding yellow plants had a coin with two tails. So it's not like they're not tossing the coin. They are, but both sides are the same. So all of their pollen grains, in the case of the pure breeding green, will inherit the lowercase y. All of their ovules will inherit the lowercase y. And in the case of the pure breeding yellow, all of the pollen grain would inherit the capital Y, all of the ovules will inherit capital Y. And every time that these pollen grains and ovules fertilize with one another would produce a new generation with those tricky coins with the same size. So the coin, quote unquote, coin model still worked for the F0, or for the pure breeding lines. It also worked for the F0 cross between pure breeding lines. Why? Because when he's crossing pure breeding lines, he's crossing plants that, again, have these loaded coins with both sides the same, with both faces the same. So it's not like these plants weren't tossing the coin. They were tossing a coin that would always give the same outcome. The pure breeding green pea producing plant would toss the coin and make an ovule cell with a lowercase y. The pure breeding yellow pea plant would toss the coin, make all pollen grains with capital Y. And that means that every time a pollen grain and an ovule fertilize between these two lines, he would get a hybrid where both traits were present, except that the dominant one is masking the recessive. All plants were like that, and not only were all the plants from this particular cross like this, if he flipped the quote-unquote sexes of the gametes that he used, so if he started using pollen grains from the pure breeding green and ovules from the yellow pea pure breeding line, he would still get 100% of the progeny being hybrid and showing the dominant trait. And then, of course, we already talked about this, that the F1 was like two tosses of real coins with different faces, and that the outcome would be all possible combinations with two heads, one tail, one head, and two tails. I'm not going to refill all of this. You can do that later. But this is the sort of the starting point for his model. Okay? Did that make sense? Any questions, anyone? Yeah.
So this is like the 3-to-1 ratio. And then
No, I welcome the question. So it's a 3-to-1 ratio, but it's also a 1-to-2-to-1 ratio. How can it be both?
because one of those two yellow plants is a hybrid.
Two of those three yellow plants are hybrids, and only one is pure breeding. So it's very important. So when we talk about these hybrid crosses, and we'll get a little more technical and call them monohybrid crosses because it's a hybrid individual for one character. So they're monohybrid. Later, as in a couple of weeks, we'll talk about dihybrid crosses. But today we're talking monohybrid crosses. We often hear a monohybrid cross produces a 3-to-1 ratio. But I could say, well, no. A monohybrid cross produces a 1-to-2-to-1 ratio. It's both. It's just that we need to be careful about what type of ratio we're talking about. It is true that it produces a 3-to-1 ratio, but this is a phenotypic ratio. What we see from outside of the plant. What color is that pea? What color is that flower? What shape is that pod? And so forth. The 1-to-2-to-1 is a genotypic ratio. We're not looking inside of the plants to see what genes they have. Mendel didn't have those tools. Most of us don't have those tools today. We can't really look inside of the plant and see what genes they have. But by the outcome of their self-fertilizations, we can tell if they're pure breeding or hybrids, right? So a monohybrid cross will always produce a phenotypic ratio of 3:1, 3 dominant for every 1 recessive, and a genotypic ratio of 1:2:1. Both things are true. Yeah? Yes?
When Mendel self-fertilized these plants, did they have all of these, like, say if it was an F1,
Uh-huh.
would all of the F2 generation come out at the same time giving that phenotype? Because I'm thinking like with childbirth, if you're thinking boy or girl, like
Uh-huh.
every time you have a child,
Uh-huh.
even if it's supposed to be a 50-50 ratio, you can have 2 girls, you
Yeah.
can have 2 boys.
Yeah.
So how did he observe the 3-to-1?
Excellent question. So it comes down to numbers, right? Whenever he took that brush, collected some pollen, he was collecting hundreds of thousands of pollen grains in the tip of that brush, introducing them into the ovary of the flower, and the ovary of the flower has tens of ovules in it. So when those seeds germinate and give rise to new plants, we're not talking 1 or 2 or 3 or even 4 plants. We're talking in the 30s and 40s. And that's where numerically he could be confident about these 3-to-1 ratios. Anytime we talk about these 3-to-1 ratios, we're not saying 3 plants to 1 plant. These are ratios. So more likely it was more like 9-to-3 or 12-to-4 and so forth. Does that make sense? Also related to your question, yes, all of these plants came out more or less simultaneously. That's something that plants and animals with very large broods can give you, right? With humans, you can't have 12 babies at once. But these ratios you can observe over time. So a lot less nowadays, but there was a time not that long ago that families would have 9, 10, 11 kids. And over time, all of that progeny came from these parents, and you could easily start to see some of these ratios and be confident about those ratios from a mathematical point of view. Yeah? Your question will come to haunt us in a way next week. We'll address it head on. Any other questions, anyone? All right, so a little bit of modernizing the nomenclature and the language. At the time of Mendel, the conclusion from his experiments, and in a way, the way that he formulates his first law, the law of character segregation, is during pollen or ovule formation, different traits for a character are separated from each other and have equal probability of being present in either the pollen or the ovule, i.e. there is equal segregation of traits into pollen or ovule. And then following fertilization, these traits combine and result in a given visible trait, which will depend on how some traits dominated over others, right? That's the language that Mendel used. Today, we've modernized this language, and more generally speaking, we say during gamete formation, because now we're bringing in animals with their sperm and egg cells, different alleles, so we can get a little more technical. Instead of talking of traits, we can talk about alleles for a locus or gene, are separated from each other and have equal probability of being present in a gamete. Again, we're being more inclusive. There's equal segregation of alleles into gametes, and following fertilization, these alleles combine and result into a given phenotype. Mendel didn't have that language. He didn't speak of phenotype. He talked about traits and characters. So characters were flower color, pea shape, pea color, sort of the broad category of the physical characteristic that he was looking at, and then the actual trait was the actual physical manifestations of those things. Green or yellow, tall or short, wrinkled or smooth, and so forth. Purple or white, that you're gonna get sick of the purple or white, I promise. We've touched on some of these already, so I'm gonna go through this quickly. Nomenclature matters, right? So you may find in questions the word character versus trait. Again, character is the type of physical characteristic versus trait, which is the actual physical manifestation of that character, right? In more modern terms, we would refer to characters as a given gene or a locus, the plural for locus, loci. And then each individual trait would be coded by a given allele, right? So we talk about alleles for the locus, flower color, or alleles for the locus, pea color, or alleles for the locus, pod shape, and so forth, right? I'm guessing that most of you are familiar with this terminology, but instead of talking about pure breeding individuals and hybrid individuals, today we will talk about homozygotes or heterozygotes. So homozygotes are those that carry two alleles of the same kind for a given locus. Again, so far we're talking about one particular locus at a time. There'll be a time soon that we're talking about two or more, but for now we're focusing on one locus. So homozygotes are individuals that carry two alleles of the same kind. We need to specify homozygous recessive or homozygous dominant. Heterozygous means one of each. In that case, it is a little silly to specify recessive or dominant because you're basically saying it's one of each, right? And then there is a term that is a little less common and probably a little less familiar to most of you, hemizygous. And that happens when instead of there being two alleles for a given locus, there is only one. That can happen in cases where there has been an aneuploidy, so a given individual or cell is missing a chromosome. So instead of having two alleles for a given character or locus, it has only one. Or in the case of, as we will see next week, sex chromosome linked genes that live in sex chromosomes, which depending on the species you may have only one chromosome of a given type, and therefore the alleles in those chromosomes will be present in only one copy. Down here, I have a little disclaimer about the way that we denote recessives and dominants. There is the classic nomenclature that Mendel introduced, capital letters for dominant alleles, recessives in lowercase. But nowadays, and in part because in time, aside from the pea plants that Mendel worked with, when you look at more and more organisms, you soon realize that many loci will have more than only two alleles, right? So, and many of them may be recessives, many of them may be dominants, depending on what combination they are. So this whole idea of capital letter and lowercase letter becomes useless. So the nomenclature we use today is a letter or a combination of letters and a little plus sign to indicate either a dominant or more often than not simply a wild type copy, a normal copy of that particular allele, and then various super indices, numbers or letters or combination of both to denote each of the alleles that we superscripts the alleles for a given locus. Okay? In this class, you may encounter examples of both. Examples in which we use the classic nomenclature of lowercase, uppercase, but then every once in a while you may encounter questions in which you see a given name for a gene, often in more than one letter, and these superscripts to indicate the various alleles that exist for that particular locus. Okay? Any questions about that? And now that we mentioned wild types, if I ask you, when thinking of that green pea versus yellow pea character or trait for the pea color character, which one was the wild type, which one was the mutant? Mutant was green and yellow was the wild type. Does everybody agree? Huh? I see a no. Why no?
I'm not sure we can tell.
I thought you had said no. You could have taken the credit. Sorry.
I'm not sure we can tell because there's no guarantee that a mutation is going to be recessive or dominant. Right?
That's one way to put it. Yes. So there's no guarantee that a mutation will be recessive or dominant. Maybe the mutant is the yellow. It's just that it's dominant over the green. So maybe the green is the wild type and the yellow is the mutant. Or maybe the green is the mutant and the yellow is the wild type. What led you think, and others, what led you to think that green was the mutant?
Because green was very recessive in all of the progenies that we did compared to the yellow.
To the yellow, right? So we have these very natural tendency to think, well, whatever more common trait I see in nature, I'm sure that's the wild type. That's the natural type. That's the type that I would find in the wild. Anything that is different from that would be a mutation. Except, we talked about this already, so it'll come back. When we're thinking about, in this particular case, different traits that you might find up in a population, we have to be careful about thinking of them as mutations. They can simply be natural variants. And that is what happens with these pea plants. The green and the yellow, we can't really say that one is a wild type and the other one is a mutant. They're both natural. They're just different variants of this particular character, right? Same with the purple versus white flowers, wrinkled versus smooth peas, and so forth. If you guys remember, we define mutants as things that are extremely rare in a population, or things that we know for a fact are harming an individual in some capacity. But in this case, there was nothing harmful about producing green peas. Those plants were totally fertile, totally fine. It's just that the green pea trait was recessive to the yellow pea trait, but that didn't mean that the possessing those green pea alleles was harmful to the plants. In fact, as far as we know, mathematically, their presence in the population was just as likely, the presence of the allele, that is just as likely as that of the yellow pea allele. It's just that when together, the yellow physically, phenotypically dominated over the green, masking its presence. But it's still there. It wasn't doing anything harmful to the plants. So that green pea trait was not a mutation in the sense that it wasn't causing any harm, and it wasn't super rare in the population, right? So we would call it a variant. And when we first talked about this, we said that anytime there was a character with many different variants, we would call those polymorphic, right? In this case, the characters that Mendel worked with were all dimorphic. They all came in two different flavors. Story goes that he actually tried or checked 32 different characters, and that there were some that were more than dimorphic, had more than two traits related to them, so that made the experiments and math a little complicated, and that some of them didn't show this distinct and clear-cut 3-to-1 phenotypic ratio and 1-to-2-to-1 genotypic ratio, so he kind of swept it under the rug because he couldn't explain it all that well. But that's how the story goes, that he actually looked at 32 characters, only 7 of them show these features. Today, one might rush to the conclusion, oh my God, he faked the data, or like he hid evidence. Now, more often than not, as scientists, we sometimes encounter these more complex than we can handle situations, and we simply stick to the simpler, the stuff that we can explain, try and build the models, and then build up from there. And that's exactly what happened with Mendel, because he started with this very simplistic, clear-cut, linear math that he could handle, and later on, his work got picked up by others, and the basis of all of that was used to explain much more complex things that follow exactly the same rules. So as we will see starting next week, there's this whole field of genetics that sometimes people refer to as Mendelian exceptions. You may still encounter this language in textbooks and sources, Mendelian exceptions. I don't like that term very much. There is many other sources, and I like this terminology better. I prefer to call it Mendelian extensions, because they represent extensions of the groundwork that Mendel built. Okay? We good? So we're revisiting this idea, but remember, mutants are either super rare in a population or we know to be harmful. If neither is the case, then we simply refer to those as natural variants. Cool. So, what has happened between Mendel and now? The biggest thing that happened between Mendel and now is that chromosomes were discovered. Mendel never saw chromosomes because he didn't have access to the microscopes that would have allowed him to see the chromosomes, nor the techniques for preparing samples that you need to see chromosomes. But other people were working on microscopy, looking at cells, and they started noticing there were these things in the nuclei of cells, of certain cells, dividing cells, that showed all of the same features or all the same characteristics of what Mendel had proposed for his so-called units of inheritance, right? So Mendel put together this idea that what parental individuals were putting in the gametes, he didn't call them gametes, he called them pollen or ovules, were units of inheritance. He didn't know they were genes. He didn't know they were made of DNA. He didn't know that those things lived in chromosomes. He didn't know any of these things. It's just that, bless you, mathematically, he knew that every time a parent produced a gamete, it was putting into these gametes units of inheritance. And when people started seeing chromosomes, they started noticing the analogies between chromosomes and Mendel's units of inheritance. First, chromosomes showed up in pairs, just like Mendel had proposed for the units of inheritance. People could see through the microscopes that in cells there would be always pairs of chromosomes that looked alike. Second, they could see that if they looked into the sexual organs of either animals or plants, they could catch cells in the act of meiosis, of making gametes, and they could see how these pairs of chromosomes started separating from each other, right? Just like Mendel had predicted for the units of inheritance. And finally, they knew that following fertilization, the chromosomes that were present in the gametes, plant or animal gametes, would combine and be present in the zygote. So chromosomes that these people could see through microscopes were behaving exactly like Mendel had proposed for the units of inheritance, which led to the famous the theory of chromosomal inheritance. And the thinking was, we'll revisit this, that chromosomes were like vessels for Mendel's units of inheritance. Right? They were simply physically carrying Mendel's units of inheritance. Cool. So what do we do now combining the two? Well, we know now that these chromosomes are the vessels for alleles for different loci, and we can easily explain that if we have a character, let's say character R, and we have a heterozygote individual in which one chromosome will carry the dominant allele and the other chromosome will carry the recessive allele. Those chromosomes will duplicate their DNA prior to meiosis, and then in meiosis 1, the pair of homologous chromosomes carrying that R locus will first face each other, then separate from each other during anaphase 1, taking with them either the dominant allele or the recessive into the daughter cells of meiosis 1. And following meiosis 2, each of the sister chromatids from each chromosome would in turn separate from each other during anaphase 2 and give rise to 4 different gamete cells carrying one allele or the other. I have a question for you. I had you kind of review meiosis. I know some of you may have reviewed it for the test, but also there was a little quiz in preparation for today to review meiosis. Knowing what you know about meiosis, what is missing from this picture here? You want to say it out loud?
Crossing over.
Crossing over, right? What is crossing over?
Like, I know that it happens.
Exactly. Physically, we say crossing over. All of us think that, right? The arms crossing. That's exactly right. So crossing over is a process by which chromatids from homologous chromosomes will cross over each other and physically interchange bits of chromatin such that now these chromosomes have swapped the allele they had. So we go from a chromosome that had 2 copies of the dominant and another chromosome that had 2 copies of the recessive to 2 chromosomes that have a copy of each. You guys see it? Yeah.
When the crossing over happens, does the telomeres as well cross over?
Yeah, yeah. And actually, we'll see that a little later. More often than not, there is several crossover points throughout the length of a chromosome arm. So sometimes what happens is there may be 2 crossover points, one here as the one shown, and then another one here. So what that does is it ends up bringing the telomere back into its original position. But any crossover point that happens, be anywhere in the chromosome arm and the telomere, will carry the telomere with it. Yeah. So how do you guys think this affects what I just said about equal segregation of alleles? Because the equal segregation of alleles kind of depended on both dominants going into one cell after meiosis 1 and both recessives going into the other cell after meiosis 1. So how will these crossover events affect that? Do you think it affects the segregation of alleles into gametes? Yes. How? That's your fault for expressing human emotions. Like, you could be a statue. Otherwise, I'll call you up.
Well, I guess because now the gamete has a copy of both the dominant and the recessive. It doesn't just have the dominant.
One or the other, right? Super intuitive. Except, here's what happens. It is true that after meiosis 1, you haven't separated the dominant from the recessive because each cell after meiosis 1 inherited a copy of each. So these alleles have not segregated from each other fully after meiosis 1 if there was a crossover event. But what happens after meiosis 2? After meiosis 2, the outcome, from the point of view of the alleles, is the same. Half of the gametes inherited the dominant, half of the gametes inherited the recessive. From the point of view of the allele, the chance of each allele to end up in a gamete is 50-50. I want you guys to notice something that is super important. On the left side of this slide, we have what gametes would look like when it comes to this particular chromosome without crossing over. And you can confidently say that there are 2 gametes that are identical to one another, 2 gametes that are identical to each other, and both of these pairs are different from each other, right? So you have 2 of one kind, 2 of the other. On the right half of this slide, we have 4 gametes that are different from one another. So what I'm saying is not that crossing over does not affect gamete variability. It actually does. Thanks to crossing over is that you can generate 4 completely different gametes. You look at these 4 cells on the right side of the slide, and they're different from one another. One has a full red chromosome, one has a blue, one has mostly red with a tiny bit of blue, and one has almost all blue with a tiny bit of red. They're different from one another. But from the point of view of the particular R locus, 2 of them have the dominant. So these 2 have the capital R, these 2 have the recessive. Crossing over will increase the variability and diversity of the gametes formed by an individual prior to sexual reproduction, but it does not affect the equal distribution of alleles into them, right? So the chances of a given gamete to have either the dominant or the recessive is still 50-50. Okay? We good? All right. So to finish this up, we have presented you with this scenario. We have a plant. This is not Mendel's pea plants, different plant. So we have a plant that is homozygote dominant with red flowers and a plant that is homozygote recessive with white flowers. What color will progeny from these plants be, which based on what we discussed so far will be heterozygote with 1 dominant and 1 recessive allele? What do you guys think? Red?
Red and white.
Red and white?
It depends on
Which?
like ratio.
Let's take a look at this again. So these are homozygote dominant, these are homozygote recessive. So what kind of gametes can these homozygote dominant produce?
Red.
Well, the gamete wouldn't be red or white. The gamete has one type of allele, right? And which type of allele would these gametes have? Always the dominant, because that's the only thing that a pure breeding, a homozygote plant can put into the gametes. Likewise, the homozygote recessive can produce only gametes with the recessive. So all of the progeny from these plants will be heterozygote. All of them will have a dominant allele and a recessive allele. So what will be the phenotype of those plants? Red flowers or white flowers? I'm hearing mostly red, right? Yes, except here's where we start getting into the extensions, the Mendelian extensions. That is true if red is fully dominant over white. Mendel's plants, when it came to flower color presented full dominance. Purple dominates over white, and in a heterozygote plant, the flowers would be as purple as in a homozygote dominant plant. You couldn't distinguish a homozygote dominant plant from a heterozygote because purple fully dominates over white, and those heterozygotes showed the full dominant phenotype. But there are cases in which you might get an intermediate phenotype, and we call that incomplete dominance, right? So if red was incompletely dominant, notice that I'm not saying that it's not dominant, I'm saying that it's just incompletely dominant over white, what you'll see in the heterozygote is some sort of intermediate between the two pure breeding phenotypes. And then there are cases of co-dominance in which, and this is a little stranger, co-dominance is when both phenotypes manifest physically, phenotypically, simultaneously. And the example that it gets thrown around a lot is blood type A codes in blood type. In individuals with blood type A, the red blood cells are coated with one sugar. Individuals with blood type B, red blood cells are coated with a different sugar. And in those individuals who have a blood type AB, both sugars are present in those cells. Nothing we could see with our naked eye, but if you look with biochemical techniques, you would see both sugars in these red blood cells. Okay? So incomplete dominance is one of those Mendelian exceptions or extensions that we need to keep in mind when we're thinking about Mendelian genetics, because it's no longer as easy as to think, okay, in the heterozygotes, the dominant allele will dominate fully over the recessive, and we will see the dominant phenotype. It can get tricky at times when there is incomplete dominance, and now the heterozygote phenotype is somewhere in between both homozygotes. And last but not least, a couple of concepts. First, the concept of penetrance. So let's say that we're crossing up, we're back to Mendel's plants. We're crossing a pure breeding purple flower plant to a pure breeding white flower plant, a homozygote recessive. We expect the F1 progeny to be heterozygote or hybrid, and all of them to have purple flowers. But then let's imagine for a second that one out of however many plants has white flowers. So that can happen. People started seeing these things, and that led to the concept of incomplete penetrance. That's when fewer individuals than expected show a predicted phenotype. And the way we calculate penetrance is by simply doing a ratio between the number of individuals that show the predicted phenotype divided by the number of individuals that you expected to show the predictive phenotype. In this case, it would be 6 divided by 7. And I've been instructed by my kid to, at this point, go 6, 7. None of you had your phones out, so this moment will be lost in history. All right, let's get back. Another concept is expressivity. I know we're running a little over, but I'll finish within a minute, I promise. That is when you expect individuals to present a given phenotype, and then you see that they present that phenotype, but there is a little diversity in the intensity of that phenotype. So for instance, the intensity of that purple color is diverse in this progeny. So we say then that this phenotype is not fully expressed. And one of your books has this interesting combination of scenarios where you can have complete penetrance and narrow expressivity, so all of the individuals show the same intensity of phenotype. You can have incomplete penetrance with narrow expressivity, so individuals may show the phenotype or not, but when they do show it, it's intense. Then you can have complete penetrance with broad expressivity, so all of them show the phenotype, but different intensities. And then incomplete penetrance with broad expressivity. Not only do not all individuals show the phenotype, in addition to that, those that do show the phenotype show different intensities of it. And all of this comes down to something that we mentioned before. That phenotype is really the combination of the genes that an individual carries, certain environmental inputs that may have nothing to do with genetics, and the combination or the interaction between the genes that an individual has and the environment. So these intensities in flower coloration or intensities in any other phenotype that you're thinking of could be a combination of the genes that you carry plus environmental inputs.